Thank you. Good afternoon. Thank you for the opportunity to talk about our studies in South Africa and Uganda on home-based HIV counseling and testing and facilitated referral to care. So antiretroviral therapy, ART, at an individual level reduces morbidity and mortality and also reduces HIV transmission by more than 90%. Modeling studies, including those discussed at this meeting, have predicted that at high ART coverage, the incidence of HIV within the population can be reduced. High coverage and retention in the testing to sustained viral load suppression cascade are essential for successful treatment as prevention. Home-based counseling and testing is a platform that has been successfully used to achieve very high coverage of HIV testing, but it needs further evaluation to see whether it's a platform that can also be used for high linkages and retention to HIV care. So this slide shows the first demonstration of home-based counseling and testing as a successful strategy for HIV testing, and the study was done by our colleague, Dr. Elioda Tumwesije, in southwestern Uganda, and was done through the Integrated Community-Based Interventions Group, ICOBI, and within the study, 89% of adults, more than 260,000 people who lived within the district were tested and received their HIV results between 2004 and 2007. So demonstrating here that HBCT really has the potential to result in universal testing and knowledge of HIV status. However, in this first study, just 11% of HIV-infected individuals were linked to care, so highlighting the need for better linkages to care. So using this platform, home-based counseling and testing, we wanted to move from this very high coverage and knowledge of HIV status, so coverage of testing and knowledge of HIV status, to thinking about how to use this platform for combination prevention. And the advantage of doing widespread testing of all adults in the community is that we could target combination prevention activities. So for linking HIV seropositive individuals to care, including ART, and for HIV serodiscordant couples, risk reduction and HIV care and ART, and then for high-risk negative individuals and for HIV negative persons, linking them to risk reduction, and also specifically for men, medical male circumcision, and possibly in the future interventions such as PrEP and microbicides. So can we use home-based counseling and testing as a platform to increase linkages to care? And today I'm going to present the results from two pilot studies, one in Busheni in southwest Uganda, and the second in KwaZulu-Natal in South Africa. In Uganda, we partnered with ICOBI, and in this first pilot study, we introduced follow-up visits at 1, 2, and 3 months to increase the linkages to care, and also introduced referrals to medical male circumcision. The second pilot study in KwaZulu-Natal introduced home-based counseling and testing to an area where it hadn't been done before, and also built on the pilot by adding point-of-care CD4 testing and viral load assessment at baseline and 6 months. And the objectives of the study were to evaluate the acceptability and uptake of HBCT, and then to evaluate the stage of HIV, and we did this by flow cytometry CD4 in Uganda, and then point-of-care CD4 in KwaZulu-Natal, validated using flow cytometry, and also a questionnaire about clinical symptoms. The third objective was to estimate the feasibility and impact of active follow-up to increase linkages to ART. And then in KwaZulu-Natal, we added 2 additional objectives at 6 months to look at viral load suppression, retention, and participant evaluation of home-based counseling and testing, and then finally to look at the incremental costs and cost effectiveness of home-based testing compared to facility-based testing. And just briefly, this slide shows the study design following community sensitization and consent. Individuals received confidential, individualized, private counseling and HIV testing. Partners were given the opportunity to do facilitated disclosure. HIV-negative individuals received counseling and referral to effective prevention strategies, for example, male circumcision. And HIV-positive individuals were referred to care, including HIV, and these individuals had follow-up visits. So once you were done with the consent, the whole process took about 20 minutes, including collecting the data, which we did on mobile phones, and actually that data collection took less than 5 minutes. So this slide shows where the study in Uganda was, in Busheni in the southwest. And the prevalence in this area going in was estimated by previous studies to be about 6%. And this slide shows very importantly, the prevention officers with their HIV testing kits and the cell phones on which we collected the data, and shows the results from the pilot. So in the Uganda pilot between November 2010 and March 2011, 855 households were enrolled. There were just over 1,900 adults living in the house, and just by visiting every household once and testing all the adults who were available, 80% of individuals, 1,558, were tested for HIV and received their results and had CD4 testing. 47% of these individuals were male. The HIV prevalence was higher than we expected, 9.8%, and the median CD4 count was 467, so much higher than is seen at voluntary counseling and testing centers. At the last follow-up visit at 3 months, 88% of individuals had visited a clinic, and 71% of those eligible had started ART. And medical male circumcision at 3 months, there was an uptake of 62%. So we wanted to build on this project, and we were able to, thanks to a PEPFAR supplement to the MP3 grant. And the aim of this additional pilot was to really test home-based counseling and testing in South Africa, an area of very high prevalence of HIV, but also when we told people we wanted to do home-based counseling and testing in South Africa, there was concerns that stigma would lead to very low uptake of testing at home. We were very fortunate to collaborate with Hedy van Rooyen and Linda Richter at the Human Sciences Research Council, and they had valuable experience in Project Accept, where they had high acceptance for home visits, about 91%, and phlebotomy for HIV testing. But they hadn't actually done the HIV testing in the home, and they hadn't done the linkage to care part of the study. We also incorporated point-of-care CD4 testing in collaboration with Dr. Wendy Stevens at the University of the Witwatersrand. And linkage to care and ART was the primary outcome of interest in this pilot. So these are the beautiful rolling green hills of KwaZulu-Natal, and in the study area of Vulindlela, the population is about 400,000. It's an area characterized by very high rates of unemployment, and the formal employment is mostly in forestry. There's a high prevalence rate of HIV, 30% in antenatal clinics in 2001 and 43% in 2004. And these are the prevention officers out in the field, going from home to home with the HIV testing kit, sometimes hiking up to houses, and then within the home, developing excellent rapport with the study participants, transforming the home into a testing center, doing the appropriate counseling and referral. And this is the team of people at our study training last year. So in South Africa, we enrolled between March and August 282 households and tested 673 adults, and that was 91% of all adults who were resident and able to provide consent within all the households that we tested, and we blanket tested the enumeration area that was defined for the study. We identified 201 HIV-positive individuals, a sobering HIV prevalence of 30%, of which 74, 37% were newly identified. It was good to see that on enrollment, 64 of the 201 participants were on ART, but just 3 of the 17 participants who had a CD4 count of less than 200, so 18% were on ART. And again, we found a very high median CD4 count at baseline, 425 cells. And indeed, just 33% of individuals had a CD4 count of less than 350, so under current guidelines would qualify for ART in South Africa. So 67% of individuals need strategies to link and retain them to care until they're eligible for ART in this setting. We found very good agreement between the point-of-care machine, and this is the instrument here shown on this slide, and it provides a CD4 count result within 20 minutes. So here showing the correlation in the first graph, and then the Bland-Altman plot showing the mean difference between flow cytometry, the gold standard by fax caliber, and the point-of-care CD4 test. The mean difference was just 16 cells, so not something that would be clinically significant. And just a few individuals, depending on the ART guideline, would not initiate ART based on the CD4 results. But this is using a very strict cutoff for CD4. And we know in clinical practice that if someone is slightly above the guideline, they probably would be initiated on ART. And this is just to show the size of the machine, that the CD4 count machine you can see on this slide that was carried from home to home. So at 6 months, we found very high linkage to care. 187 of the 196 people who were eligible for a 6-month visit went to the clinic, so 95% of people linked to care. And 35 individuals of those of people who are not on ART, 26% initiated ART. For those people who had a CD4 count of less than 200, 11 out of the 13 who were eligible initiated ART, so 85%. During the course of the study, the ART initiation guidelines changed from a CD4 of equal to or less than 200 to a CD4 of equal to or less than 350. And this slide shows the testing to viral suppression cascade that we've seen several times at this meeting. And the first graph shows the coverage that we found at baseline. So of the 203 individuals, in fact, many of them, 120, actually had a CD4 count taken, but very few, 70, had gone back to get the results. So that shows the first 2 drops in the testing, the first two losses to retention in the testing cascade in the first graph. But of those people who had a CD4 count and went to clinic and were eligible, many of them, 96%, initiated ART and were virally suppressed. The next slide shows the cascade after the home-based testing. And here we can see, because we did point-of-care testing, we identified everyone who had HIV, everybody, 100% received their results and was staged, so received their CD4 count results. 95% of people went to the clinic and were linked to care. And the drop after that is due to the ART initiation guidelines at the clinic, but by 6 months, 88% of those eligible initiated ART. So we found that home-based counseling and testing is acceptable and can achieve very high coverage, more than 90%. It identifies HIV-infected individuals who were previously unaware of their status and early on in their infection. There was very good agreement between the point-of-care test and the flow cytometry CD4 results. And the point-of-care test was highly acceptable. It saved patient costs in returning to the clinic 2 or 3 times in order to get their CD4 results. And we were able to do specific counseling based on their CD4 results. So we were able to counsel people about whether they were eligible for ART and what the next steps were. So we found home-based counseling and testing can facilitate effective linkages to care, and that the follow-up visits were successful to increase linkage to ART care and ART initiation. The next steps of the study are to estimate the effectiveness, and tomorrow Connie Kellum will be presenting a late breaker with the viral load suppression results. We're going to go on to estimate the incremental costs and incremental cost effectiveness of the strategy compared to VCT. And the ultimate aim of our work is to answer implementation science questions about the role of home-based testing in national HIV treatment and prevention programs. And specifically, we'd like to break down the components of this project to see what really resulted in such high testing and retention rates. Was it the follow-up visits? Was it using point-of-care tests? So we'd like to get at those answers a little more closely. Of course, the numbers I've showed you today are small, so we've started phase 2 of the work, both in South Africa and Uganda, a larger scale demonstration project in which up to 3,000 households will be visited. And the focus of the demonstration project is on knowledge of HIV status and facilitating linkages to care for HIV-infected individuals. So our target enrollment is 600 HIV-positive individuals with point-of-care testing, follow-up visits at 1 month, and then 3 months, every 3 months thereafter for a year. We'll measure viral load at baseline 6 months and at 12 months. And we have a nested community cohort to measure the impact of HBCT on testing, risk behaviors, and stigma, and this is funded through a NIH Directors Award. So I'd like to thank the study participants, the outstanding staff that we have at ICOBI and HRSASI, our partners in this work, all the investigators on the study team, and particularly Connie Kellum, the principal investigator for the study, and of course our funders. Thank you.